Hox genes, we know, are the subset of homeobox genes, and they are a group of related genes that specify region of the body plan of an embryo along the head tail axis of the animal. The Hox proteins encode and specify the characteristic of position. Mice and humans are the two best studied mammals, while Drosophila is the best studied insect in invertebrates. So, when we talk about mice and humans, we see that they also have clusters of homeobox containing genes, and these genes are expressed in particular segment of the animal, just like in fruit fly. In humans, there are 38 genes which are divided into 4 clusters, each located on different chromosomes. In Drosophila, the homeobox genes are arranged in a collinear manner. Similarly, in mice and humans also, these homeobox genes are arranged in collinear fashion in the same order as in Drosophila on each chromosome, as they are expressed from anterior to posterior of developing animal, as I have told you in earlier video about the temporal arrangement and temporal expression of genes. Now, one is intrigued to ask the question that what is the function of homeobox? Homeobox are present in almost all eukaryotes. The homeobox sequence encode a region of 60 amino acid, and this protein is called the homeodomain. That is the part of homeobox sequence protein. Most of these proteins are the transcription factors. They return back to the nucleus, bind to DNA, regulating the transcription of other genes. Some genes with homeoboxes are expressed only at certain times and in certain tissues as the development proceed, as would be expected if these proteins regulate development. As I have talked about the temporal expression of these genes. Then, what are we to make of the presence of homeobox in species as diverse as humans, fruit flies, frogs, nematodes, plants, etc., of its presence in several genes in same organism? The presence is consistent with the hypothesis that a single gene in an ancient organism was evolutionary progenitor of what is now widespread controlling system for development. One of the most astonishing finding of developmental biology research has been the conservation of gene involved in development pathways. Though there are certainly differences in the end product of humans and fruit fly development, but the basic genetic mechanism is similar. So, we were talking about one gene which has been propagated, multiplied several times, and appeared in different organisms. The evolution is caused by heritable changes in the development of organism. All the organic beings have been formed on two great laws. First is the unity of type, and second is known as the condition of existence. Now, unity of life means that various organisms, they have same pattern of development, same structural organization of development. This shows the unity of type. We also know that though all the organisms, they have same structural pattern, but still they differ markedly from one another. For example, if we talk about phylum Arthropoda, we have various types of animals in the group. Class Insecta have insects like butterfly, moth, Drosophila, mosquito. Then class Crustacea has varied amount of crustaceans like prawn, crabs, etc. Then we have diplopods, chelopods, arachnids, all varied and different kinds of organism. So, though there is a unity in or similarity in the structure of all these organisms, the structural organization is same. So we say that there is unity of type, but the variation is also there, and it is called the condition of existence or the phylogenetic adaptation. Darwin noted that the homologies between the embryonic and larval structure of different phyla provides an excellent evidence for explaining the descent with modification. Now, studies on Hox gene, which is said to be a, a cluster of genes, many genes, which specify the region of body plan of an embryo, which also encode the characteristic of position, ensuring the correct structure forms in correct place of the body. For example, antenna should be formed in the head region, wings and legs should be formed in the thoracic region. So the Hox genes, as I have already talked about, contains 8 genes, and these genes, they ensure that the correct structure form in correct place. The Hox gene in insects specify which appendages form on a segment. For example, leg, antenna, wings in fruit fly. And Hox gene in vertebrates specify the type and shape of vertebrae that will form in segmented animals. The Hox protein thus confer segmental or positional identity. The studies on Hox genes in ciliated larvae have shown that they are only expressed in future adult tissues. The protein product of each Hox gene is a transcription factor. Each Hox gene contains a well-conserved DNA sequence known as the homeobox. Product of Hox genes are called the Hox protein, and Hox protein are subset of transcription factors which are proteins and that are capable of binding to specific nucleotide sequence on DNA called enhancer, through which they either activate or repress hundreds of genes. This is a diagram showing the conservation of the Hox gene in various arthropod animals. In 1990, a broad agreement on the general form of a phylogenetic tree of life began to emerge among paleontologists, molecular biologists, and developmental geneticists. This consensus came from first, improved method of using DNA for phylogenetic analysis, taking into account its variation within group of animals. Second, new data on conserved regulatory gene sequence like Hox gene, which are stable within phyla but can diverge between the phyla. Third, morphological evidence for the related nature of some structure that had once been thought to be distinct. It is doubtful that we will ever find a fossilized representative of ancestral phylum that gave rise to both the deuterostomes and the protostomes. A hypothetical animal is referred to as the Urbilaterian ancestor. Urbilaterian. Such an animal, the Urbilaterian ancestor, had neither bony endoskeleton, a deuterostome trait, nor a hard exoskeleton, a character of ecdysozoon. The purpose is to find out the homologous genes that are performing the same function in both deuterostomes and protostomes. Many such genes have been found, and their similarity of structure and function in protostomes and deuterostomes suggest that these genes emerged in an animal that is ancestral to both the groups. Besides the Hox genes, which specify the structural positioning and the formation of different appendages from the right segment, there are other homeotic genes like the PAX6 gene, which codes for PAX6 protein. It plays a role in the formation of eye. It is very important to be present in both vertebrates and invertebrates for the development of eyes. Another genes shared by deuterostomes and protostome is a homeobox containing gene Tin Man. The Tin Man protein is expressed in the Drosophila mesoderm, eventually residing in the region of cardiac mesoderm. Loss of function or mutation of Tin Man gene lack the formation of heart. In mice, the homologous gene for Tin Man is called NK alpha 225, and it too is originally expressed in splanchnic mesoderm, splanchnic mesoderm, and then continues to be expressed in those cells that form the heart tubes. Although the heart of vertebrate and insects are different, with some common feature, that is their ability to pump the fluid. Hox gene is another homeotic gene. The expression of Hox gene provides the basis for anterior-posterior axis specification throughout the animal kingdom. Hox gene provide one of the most remarkable piece of evidence for deep homologies among all the animals of the world. The Hox gene of different phyla are not only homologous, but they are in the same order on their respective chromosome. So, the product of Hox gene is called the Hox protein. The homeobox gene and thus the homeodomain protein for Hox genes are found in most eukaryotes. Hox gene, being a subset of homeobox gene, arose more recently in evolution within the animal kingdom. Comparing the homeodomain sequence between Hox protein often reveal greater similarity between species than within the species. In most bilaterian animal, Hox genes are expressed in staggered domain along head to tail axis of embryo, suggesting that their role in specifying position is a shared ancient feature.